Kingdom, biology, the hierarchy of biological classifications. 8 major taxonomic ranks. A domain contains one or more kingdoms. Intermediate minor rankings are not shown. In biology, kingdom, Latin, renum, plural rena, is the second highest taxonomic rank, just below domain. Kingdoms are divided into smaller groups called phyla. Traditionally, some textbooks from the United States used a system of 6 kingdoms: Animalia, Plantae, Fungi, Protista, Archaea/Archaeobacteria, and Bacteria/Eubacteria, while textbooks in Great Britain, India, Australia, Latin America and other countries used 5 kingdoms: Animalia, Plantae, Fungi, Protista and Monera. Some recent classifications, based on modern cladistics, have explicitly abandoned the term kingdom, noting that the traditional kingdoms are not monophyletic, that is, do not consist of all the descendants of a common ancestor. Contents hide. 1. Definition and associated terms. 2. Modern view. 2.1 The 3 domains of life. 2.2 Kingdoms of the eukaryota. 3. Historical development. 3.1 Cavalier-Smith systems. 3.1.18 Kingdoms. 3.1.26 Kingdoms. 3.1.3 7 Kingdoms. 4 Summary. 5 Virus. 6 See also. 7 References. 8 Further reading. 9 External links. Definition and associated terms. When Carolus Linnaeus introduced the rank-based system of nomenclature into biology in 1735, the highest rank was given the name kingdom, and was followed by 4 other main or principal ranks: class, order, genus, and species. 1. Later 2 further main ranks were introduced, making the sequence kingdom, phylum, or division, class, order, family, genus, and species. 2. In the 1960s a rank was introduced above kingdom, namely domain, or empire, so that kingdom is no longer the highest rank. Prefixes can be added, so subkingdom, subregnum, and infrakingdom, also known as infraregnum, are the 2 ranks immediately below kingdom. Superkingdom may be considered as an equivalent of domain or empire, or as an independent rank between kingdom and domain or subdomain. In some classification systems the additional rank branch, Latin, ramus, can be inserted between subkingdom and infrakingdom, for instance, protostomia and deuterostomia in the classification of Cavalier-Smith. 3. Modern view. The 3 domains of life. Main article, domain, biology, a phylogenetic tree, based on raw data showing Waze's 3-domain system. All smaller branches can be considered kingdoms. From around the mid-1970s onwards, there was an increasing emphasis on comparisons of genes at the molecular level, initially ribosomal RNA genes, as the primary factor in classification. Genetic similarity was stressed over outward appearances and behavior. Taxonomic ranks, including kingdoms, were to be groups of organisms with a common ancestor, whether monophyletic, all descendants of a common ancestor, or paraphyletic, only some descendants of a common ancestor. Citation needed. Based on such RNA studies, Colway's thought life could be divided into 3 large divisions, and referred to them as the 3 primary kingdom model, or a kingdom model. 4. In 1990, the name domain was proposed for the highest rank. 5. This term represents a synonym for the category of dominion, latitudinum, introduced by Moore in 1974. 6. Unlike Moore, Willis and others, 1990, did not suggest a Latin term for this category, which represents a further argument supporting the accurately introduced term dominion. 7. Willis divided the prokaryotes, previously classified as the kingdom Monera, into 2 groups, called Eubacteria and Archaeobacteria, stressing that there was as much genetic difference between these 2 groups as between either of them and all eukaryotes. According to genetic data, although eukaryote groups such as plants, fungi, and animals may look different, they are more closely related to each other than they are to either the Eubacteria or Archaea. It was also found that the eukaryotes are more closely related to the Archaea than they are to the Eubacteria. Although the primacy of the Eubacteria-Archaea divide has been questioned, it has been upheld by subsequent research. 8. There is no consensus on how many kingdoms exist in the classification scheme proposed by Waze. Kingdoms of the Eukaryote phylogenetic and symbiogenetic tree of living organisms, showing the origins of eukaryotes and prokaryotes. One hypothesis of eukaryotic relationships, modified from Simpson and Roger, 2004, 9. In 2004, a review article by Simpson and Roger noted that the protista were a grab bag for all eukaryotes that are not animals, plants or fungi. They held that only monophyletic groups should be accepted as formal ranks in a classification, and that, while this approach had been impractical previously, necessitating literally dozens of eukaryotic kingdoms, it had now become possible to divide the eukaryotes into just a few major groups that are probably all monophyletic. 9. On this basis, the diagram opposite, redrawn from their article, showed the real kingdoms, their quotation marks, of the eukaryotes. 9. A classification which followed this approach was produced in 2005 for the International Society of Protistologists, by a committee which worked in collaboration with specialists from many societies. It divided the eukaryotes into the same 6 supergroups. 10. The published classification deliberately did not use formal taxonomic ranks, including that of kingdom, life domain, bacteria domain, archaea domain, eukaryota, excavita, various flagellate, protozoa, amoebozoa, most lobozomoboids and slime molds, opisthoconta, animals, fungi, chonoflagellates, etc., rosaria, foraminifera, radiolaria, and various other amoeboid protozoa, chromalveolata, stramenopiles, brown algae, diatoms, etc., haptophyta, cryptophyta, or cryptomonids, and alveolata, archoplastida, or primoplantae, land plants, green algae, red algae, and glaucophytes. In this system, the multicellular animals, metazoa, are descended from the same ancestor as both the unicellular chonoflagellates and the fungi which form the opisthoconta. 10. Plants are thought to be more distantly related to animals and fungi. However, in the same year as the International Society of Protistologists classification was published, 2005, doubts were being expressed as to whether some of these supergroups were monophyletic, particularly the chromalveolata. 11. And a review in 2006 noted the lack of evidence for several of the supposed 6 supergroups. 12. As of 2010, there is widespread agreement that the rosaria belong with the stramenopiles and the alveolata, in a clade dubbed the SAR supergroup. 13. So that rosaria is not one of the main eukaryote groups. 14, 15, 16, 17, 18. Beyond this, there does not appear to be a consensus. Rogers and others in 2009 noted that the deep phylogeny of eukaryotes is an extremely difficult and controversial problem. 19. As of December 2010, there appears to be a consensus that the 2005-6 supergroup model does not reflect the true phylogeny of the eukaryotes and hence how they should be classified, although there is no agreement as to the model which should replace it. 15, 16, 20. Historical development lifetime line view discuss edit -4500 -4000 -3500 -3000 -2500 -2000 -1500 -1000 -500 -0. Water single-celled life photosynthesis eukaryotes multicellular life land life dinosaurs mammals flowers. Earliest Earth 4540. Earliest water earliest life LHB meteorites earliest oxygen atmospheric oxygen crisis earliest sexual reproduction Ediacara Cambrian explosion earliest humans PHRNEROZOIC PROTEROZOICARCHERNHADERN Pangala Huronian Cryogenian and Incaru Quaternary axis scale. Millions of years ago. Orange labels. Ice ages. Also see, human timeline and nature timeline. The classification of living things into animals and plants is an ancient one. Aristotle, 384-322 BC, classified animal species in his History of Animals, while his pupil Theophrastus, c. 371 c. 287 BC, wrote a parallel work, the Historia Plantarum, on plants. 21. Carl Linnaeus, 1707 to 1778, laid the foundations for modern biological nomenclature, now regulated by the nomenclature codes. In 1735, He distinguished two kingdoms of living things, Renum animale, animal kingdom, and Renum vegetabile, vegetable kingdom, for plants. Linnaeus also included minerals in his classification system, placing them in a third kingdom, Renum lapidum. Life Renum vegetabile Renum animale in 1674, Antony van Leeuwenhoek, often called the father of microscopy, sent the Royal Society of London a copy of his first observations of microscopic single-celled organisms. Until then, the existence of such microscopic organisms was entirely unknown. Despite this, Linnaeus did not include any microscopic creatures in his original taxonomy. At first, microscopic organisms were classified within the animal and plant kingdoms. However, by the mid-19th century, it had become clear to many that the existing dichotomy of the plant and animal kingdoms had become rapidly blurred at its boundaries and outmoded. 22. In 1866, Ernst Haeckel proposed a third kingdom of life, the Protista, for neutral organisms which were neither animal nor plant. Haeckel revised the content of this kingdom a number of times before settling on a division based on whether organisms were unicellular, protista, or multicellular, animals and plants. 22. Life kingdom Plantae kingdom Protista kingdom Animalia. The development of the electron microscope revealed important distinctions between those unicellular organisms whose cells do not have a distinct nucleus, prokaryotes, and those unicellular and multicellular organisms whose cells do have a distinct nucleus, eukaryotes. In 1938, Herbert F. Copeland proposed a four-kingdom classification, elevating the protist classes of bacteria, Monera, and blue-green algae, Phycochromacea, to phyla in the novel kingdom Monera. 22. The importance of the distinction between prokaryotes and eukaryotes gradually became apparent. In the 1960s, Stanier and Van Niel popularized Edouard Chatton's much earlier proposal to recognize this division in a formal classification. This required the creation, for the first time, of a rank-above kingdom, a superkingdom or empire, later called a domain. 23. Haeckel's original, 1866, conception of the three kingdoms of life, including the new kingdom Protista. Notice the inclusion of the cyanobacterium nostoc with plants. Life domain bacteria kingdom Monera empire eukaryota kingdom Protista kingdom Plantae kingdom Animalia. The differences between fungi and other organisms regarded as plants had long been recognized by some. Haeckel had moved the fungi out of Plantae into Protista after his original classification. 22, but was largely ignored in this separation by scientists of his time. Robert Whittaker recognized an additional kingdom for the fungi. The resulting five-kingdom system, proposed in 1969 by Whittaker, has become a popular standard, and with some refinement, is still used in many works and forms the basis for new multi-kingdom systems. It is based mainly upon differences in nutrition. His Plantae were mostly multicellular autotrophs, his Animalia multicellular heterotrophs, and his fungi multicellular saprotrophs. The remaining two kingdoms, Protista and Monera, included unicellular and simple cellular colonies. 24. The five-kingdom system may be combined with the two-empire system, Life empire Prokaryota kingdom Monera empire Eukaryota kingdom Fungi kingdom Protista kingdom Plantae kingdom Animalia. In the Whittaker system, Plantae included some algae. In other systems, for instance, Margulis system, Plantae included just the land plants, Embryophyta. Despite the development from two kingdoms to five among most scientists, some authors as late as 1975 continued to employ a traditional two-kingdom system of animals and plants, dividing the plant kingdom into subkingdoms Prokaryota, bacteria, and cyanophytes, mycota, fungi and supposed relatives, and chlorota, algae, and land plants. 25. Cavalier-Smith's Systems main article, Cavalier-Smith's System of Classification 8 Kingdoms. See also, Archaebacteria, Chromista, and Archozoa. Thomas Cavalier-Smith thought at first, as it was nearly consensually admitted at that time, that the difference between Eubacteria and Archaebacteria was so great, particularly considering the genetic distance of ribosomal genes, that they needed to be separated into two different kingdoms, hence splitting the empire bacteria into two kingdoms. He then divided Eubacteria into two subkingdoms, Negibacteria, gram-negative bacteria, and Possibacteria, gram-positive bacteria. Technological advances in electron microscopy allowed the separation of the chromista from the plantae kingdom. Indeed, the chloroplast of the chromasts is located in the lumen of the endoplasmic reticulum instead of in the cytosol. Moreover, only chromasts contain chlorophyll C. Since then, many non-photosynthetic phyla of protists, thought to have secondarily lost their chloroplasts, were integrated into the kingdom chromista. Finally, some protists lacking mitochondria were discovered. 26. As mitochondria were known to be the result of the endosymbiosis of a proteobacterium, it was thought that these amitochondriate eukaryotes were primitively so, marking an important step in eukaryogenesis. As a result, these amitochondriate protists were separated from the protist kingdom, giving rise to the, at the same time, superkingdom and kingdom Archozoa. This was known as the Archozoa hypothesis. This superkingdom was opposed to the metakaryota superkingdom, grouping together the 5 other eukaryotic kingdoms: Animalia, Protozoa, Fungi, Plantae, and Chromista. 6 Kingdoms. In 1998, Cavalier-Smith published a 6-kingdom model, 3, which has been revised in subsequent papers. The version published in 2009 is shown below, 14, compared to the version he published in 2004. 27. The alveolates and the rosarians have been moved from Kingdom Protozoa to Kingdom Chromista. Cavalier-Smith no longer accepts the importance of the fundamental eubacteria-archaebacteria divide, put forward by Weiss and others, and supported by recent research. 8. His kingdom bacteria includes Archaebacteria as a phylum of the subkingdom Unibacteria, which comprises only one other phylum, the Possibacteria. The 2 subkingdoms Unibacteria and Negibacteria of kingdom bacteria, sole kingdom of empire Prokaryota, are opposed according to their membrane topologies. The bimembranous-unimembranous transition is thought to be far more fundamental than the long branch of genetic distance of Archaebacteria, viewed as having no particular biological significance. Cavalier-Smith does not accept the requirement for taxa to be monophyletic, holophyletic in his terminology, to be valid. He defines prokaryota, bacteria, negibacteria, unibacteria, and possibacteria as valid paraphyletic, therefore monophyletic in the sense he uses this term. Taxa, marking important innovations of biological significance, in regard of the concept of biological niche. In the same way, his paraphyletic kingdom Protozoa includes the ancestors of Animalia, Fungi, Plantae, and Chromista. The advances of phylogenetic studies allowed Cavalier-Smith to realize that all the phyla thought to be archozones, that is primitively amitochondriate eukaryotes, had in fact secondarily lost their mitochondria, most of the time, by transforming them into new organelles, hydrogenosomes. This means that all living eukaryotes are in fact metakaryotes, according to the significance of the term given by Cavalier-Smith. Some of the members of the defunct kingdom Archozoa, like the phylum Microsporidia, were classified into kingdom Fungi. Others were classified in kingdom Protozoa like Metamonida, which is now part of infrakingdom Excavata. The diagram below does not represent an evolutionary tree. Life empire Prokaryota kingdom bacteria includes Archaebacteria as part of a subkingdom empire Eukaryota kingdom Protozoa, for instance Amoebozoa, Chonozoa, Excavata kingdom Chromista, for instance Alveolata, Cryptophytes, Heteroconta, brown algae, diatoms etc., Haptophyta, Rosaria kingdom Plantae, for instance Glaucophytes, red and green algae, land plants kingdom Fungi kingdom Animalia. 7 Kingdoms. Cavalier-Smith and his collaborators revised the classification in 2015 and published it in PLOS One. In this scheme they reintroduced the division of prokaryotes into 2 kingdoms, bacteria equals eubacteria and archaea equals archaebacteria. This is based on the consensus in the taxonomic outline of bacteria and archaea, Toba, and the Catalogue of Life, 28. Summary. A summary of the different kinds of proposed classification schemes presented in this article is summarized in the table below. Linnaeus 1735 left square bracket 29 equal 1866 left square bracket 30 Chatton 1925 left square bracket 31 32 Copeland 1938 left square bracket 33, 34, Whitaker 1969 left square bracket 35, Weiss and others 1977 left square bracket 36, 37, Weiss and others 1990 left square bracket 38, Cavalier Smith 1993 left square bracket 39, 40, 41 Cavalier Smith 1998 left square bracket 42, 43, 44, Ruggiero and others 2015 left square bracket 45, 2 kingdoms 3 kingdoms 2 empires 4 kingdoms 5 kingdoms 6 kingdoms 3 domains 8 kingdoms 6 kingdoms 7 kingdoms, not treated. Protista prokaryote monera monera eubacteria bacteria eubacteria bacteria bacteriarchy bacteriarchy bacteria eukaryota protista eukaryoarchy protozoa chromista vegetabilia plantae fungi animalia. The kingdom level classification of life is still widely employed as a useful way of grouping organisms. Notwithstanding some problems with this approach, kingdoms such as bacteria represent grades rather than clades, and so are rejected by phylogenetic classification systems. The most recent research does not support the classification of the eukaryotes into any of the standard systems. As of April 2010, no set of kingdoms is sufficiently supported by research to attain widespread acceptance. In 2009, Andrew Roger and Alastair Simpson emphasized the need for diligence in analyzing new discoveries. With the current pace of change in our understanding of the eukaryote tree of life, we should proceed with caution. 46. Virus. There is ongoing debate as to whether viruses, obligate intracellular parasites that lack metabolism and are not capable of replication outside of a host, can be included in the tree of life. 47, 48, 49. If included, their placement in the tree would be problematic, since it is suspected that viruses have arisen multiple times, and they have a penchant for accumulating nucleotide sequences from their hosts. A principal reason for inclusion comes from the discovery of unusually large and complex viruses, such as mimivirus, that possess typical cellular genes,